Hello, everybody. Welcome, one and all. You've just tuned in with us once again for Cyberhuman Frontiers, Saturday, January 30th, 2021. All right. We have taken a little bit of a respite from our in-depth technological and biotechnological surveys and forays to take a look at some of the events of the last couple of weeks. And we will now resume our usual programming. And I use that word programming loosely. We encourage you to participate with us. And we're going to now get back into some absolutely amazing stuff that's going on. And as we usually say on these shows, some of the work that we feature and that we show and we detail is work that's been going on for a period of years. Some of it goes back 5, 10, 20 years, and we've got some presentations that we're going to play a little bit of for you and get some commentary going between you and us. And it's not brand new, but it's a couple of years old. So, but I have to say, we probably aren't particularly well informed about what's going on in those institutions of higher learning and very high-minded research, very well-financed research as well, in the areas of biochemistry and the interplay between biochemistry and electronics. There's just a huge amount of attention that's going into this, and it just keeps coming up. We don't even have to look hard for this stuff to find it, and we always have that lingering question: why such a priority on this? And well, we'll develop that thought, and we'll go back and forth as we get going in the show here. So, TMC, how are you? Are you ready?
Yeah, let's go.
All right. Well, imagine, if you will, a world in which, after millions of years of natural evolution on a planet that kept itself balanced over those eons and maintained a kind of a natural check and balance between one species and another species, and the environment and the carbon dioxide and the oxygen and the sunlight and the warmth and the seasons and all that. Imagine, if you will, a world that has certainly passed the test of time in keeping itself operating, that we were to come along in just the last few years, developing abilities and technologies that could literally catapult the shape of life and the process of what had previously been millennia way, way, way ahead all of a sudden. And the thought that I put to you is, would this not be potentially something alien to the natural earth? Would this be something that is developed, designed, built by the minds of man and woman, that is injecting something new and radically different into our biosphere in an extremely short period of time? Now, excuse me, we don't need to get into the areas of creationism versus evolution, anything like that. All that we really have to do is take a look at the fact that we do have some sort of a historical track record, a long-term historical track record of the development of some life forms on this planet, and many, many, many fossil records that show those developments. And all I'm trying to establish with that is that there's been a lot of time that has elapsed, and change in the natural world takes place gradually. So here comes along man, doop, 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 designing stuff in a biochemistry laboratory and building things that never have been seen before. New designs, new gene splicing, which blends together genetic traits of things, for example, such as the dye in a squid with a mammalian life form, or blends together the type of proteins that make the web that a spider uses with a goat, and many, many other examples of genes that have been mixed together. But now we're into and onto even a further advancement of the frontier, which is there's a drive in these research institutions to find a way to develop, well, I guess the only way to put it is biological wiring, biochemical wiring. So this biochemical wiring has as its end result being able to interface a biological creature such as ourselves with outboard electronics. And there's a real push for this stuff. And it absolutely fascinates me, but it does scare the crap out of me a little bit because we really don't know where that's going to lead. But it's going on, and we're going to show you. And so I get back to that point that I was trying to make. Would this indeed would it be valid to classify this as something alien to the general developmental track of life on Earth? And I think that, I guess you could say that it's natural if we regard the development and the engineering that humans do to be a natural outgrowth of the evolution on Earth. Then you could say, well, all right, so this life form came along and it's really smart and it knows how to, do things a little bit more advanced than be a beaver making a beaver dam, or follow instinctive rules like salmon and swim upstream to spawn. It's now building new unseen combinations of life characteristics from various genomes that have now been cataloged and categorized and can be combined using gene editing and gene splicing technology, such as, for one, CRISPR. But there are others. So I just say, I haven't even answered the question. Are we becoming alien to the Earth that has been our home and our the repository of all life for so many millennia? Are we becoming the alien creatures? It really gets me going. Or no, it's just us. Here we are. We may carry it so far that we could perhaps extinct all of ourselves by accident if things get away from us. But it is definitely a brave new world. Science fiction comes true. And what we want to fascinate and amaze yourselves and ourselves both together tonight as we look at some of this stuff. And then in the back of our minds, the question, how might this change our lives, our environment, our future? It will. I guarantee you, we're certain of it. It is definitely going to be changing the future direction of humanity. We're going to be reshaped. We're going to be changed somewhat, and hopefully it's not at the hands of our own fatality. We are on the verge. There are many potential outcomes. Some of them are absolutely stupendous, amazing for the improvement of the human condition as well as for the rest of the natural world. And others could be, they could be a little bit darker than that. So we're going to set the foundation. We're starting on a new group of topics, and with your fearless leader TMC there, deciding which area of this we're going to investigate and survey. I think that you're going to be fascinated. We want you to stick with us for a few episodes as we go through this. And so anyway, going to start off. TMC, do you want to add anything before I start off with just a couple of little graphics here?
No, not really. I think that we've been through this cycle before, and it didn't end well. So,
yes, that has been suggested. Yeah, that's really been suggested, and we see evidence of ancient technologies that seemed so far advanced from what we would have expected, but there they were. I'm going to put up a share now, and you'll all be happy to know that I did some updating on my computer. I might or might not get my echoing. Now, am I echoing? Am I echoing? No echoing?
Mm-hmm.
Cool. That's good. All right. So I got this picture up on the screen. And what we're looking at here is we are looking at cellular structures superimposed upon our digital schematics. All right. So this just kind of gives you a little bit of an idea what kinds of things we're talking about. And there are people who are doing intensive research on taking some of these circuits. And what we have here is a logic gate. If you can see, let me take a look on the share and see if you can see that. Yeah, that's a logic gate. That's another logic gate. And they are being controlled by this input right here going over there, and then output from some sort of a processor or an interface device. And I think that what there is here, I can't really make it out too well, but I can tell you that there's going to be a connection to the outside the cellular structure, either to another part of the organism or perhaps out to an electromagnetic interface to computer equipment. So this is the type of thing that's being designed and worked on. And I'm going to come back to this because this is part of an article, and we can read at our convenience. We'll see how things flow. But now, for added amazement, we're going to move over to this. And before I actually begin playing anything, I'm going to ask for your patience while I read our community guidelines disclaimer. The points of view and purpose of this video is not to incite fear, violence, harass, or bully in any way, but to share opinions and thoughts with other like-minded individuals curious about the subject, to encourage conversation and awareness. The opinions expressed on this stream belong solely to the persons espousing them and are not an expression of the channel Cyberhuman Frontiers. This video does not claim to give medical, legal, political, or financial advice. And this video and our YouTube channel in general may from time to time contain certain copyrighted works that were not specifically authorized to be used by the copyright holder, but which we believe in good faith are protected by federal law and the fair use doctrine for one or more of the reasons noted above in the Copyright Act of 1976. Thank you very much for that. And now we're going to move, I just grabbed the wrong mouse, to this, what I would suggest that this is a brilliant lady who has done a whole lot of work and a whole lot of study. And we're going to listen to her a little bit, and she's going to talk, as you can read on the screen, going to talk about electronically controlled microorganisms and synthetic biology. What we mean by synthetic biology is we mean designing amino acids and proteins that did not exist in a normal, unmodified in vivo organism, but have been changed so that I'm going to suggest that they're being changed for the purpose of being able to have that electronic interface with the organism. Now, they're doing this work with microorganisms, but of course, you can always look forward and scale up. You know, it'll be going to more complex organisms as it goes forward. So we would like to thank in advance Carolyn Ajo Franklin. And this is one of the DeLange conferences. And this is from a couple of years ago. And I didn't know this stuff was going on, but it's very interesting. And here we go. Pick a good place to start. Right about here. Okay. And TMC just flag me if the audio's too low or too high. All right. We're going to let her run for a good few minutes because I want you to get the feel for where she's going. And I will intervene after just a couple of minutes to point out a couple more things.
Great privilege to be here at Rice University, and I really have to thank the organizing committee for not only putting this very interesting discussion together, but actually for the incredible hospitality that they've shown over the course of the meeting. So I want to talk today about how we are using synthetic biology to create electronically controlled microorganisms. And I want to kind of harken back first to why we're doing this. And I had the great privilege of working with Pam Silver as a postdoc. When she was starting up the synthetic biology effort in her lab. And I think, as you'll see from this picture, Pam really inspired me and many others to really follow in her wake and to think about how synthetic biology could really tackle the challenges of the 21st century. And so I think one of the biggest challenges in the 21st century is the realization that we are changing the planet on an unprecedented scale. We kind of have actually, geologists have actually named this new era the Anthropocene.
And so just trying to help everybody to go along with my thought process, which you can reject if you want to think in a different direction. But she's making a case that something needs to be done, calling this the Anthropocene age, and that there is a whole lot of anthropomorphogenic change happening on the planet, which I think isn't debatable. I think that's true. But this is scientists coming to the rescue. And I always go back, in the back of my mind, I think, okay, thousands and thousands and thousands of years of the Earth just tootling along just fine. The bees and the birds were happy, the plants, the animals, all that stuff. And along with them, there were humans too. But anyway, maybe this is the way that we should be going. It certainly looks like that's the way that we are going. I took the liberty to speed this up to 1.25x because we have a lot to cover. If that's too fast, somebody let me know.
The incredible changes that humans are having on the surface of the planet, the chemistry of the planet, the microbial diversity, and indeed diversity of life on the planet. So when I talk to colleagues in the plant and microbial biology community, one of the things they'll emphasize to me is that we actually are on the verge of potentially the 6th mass extinction event across the world. If we look at the rate at which we are losing organisms, species, it's 10 to 100 times faster than the kind of normal rate. And
TMC, I can't contain myself. She makes a good point, and I think that she's right, but okay. What is the reason why that's happening? It's because of all of the modifications, the willy-nilly modifications that humanity is doing on our planet, making plastics, ejecting them out into the ocean, shooting radioactivity up there, all kinds of stuff like that. So we have scientists coming along to rescue what other scientists did, and so isn't there some kind of an approach where you just back off and say, well, wait a minute, let's stop screwing with it so much and let nature reorganize itself and purify itself, which it does on a regular basis. And by the way, hi, everybody out there. I see you all. Good to see you, Truth Hunter and Powder KLC, everybody. Good to see you out there. Do you have any just quandaries or thoughts, or should I just let her go a little further?
I think that she's implying that
we need to fix it.
No, through the course of trying to understand, master, and ultimately create what it is we think we need that isn't already here naturally, we could create something that would cause a great deal of destruction. So like a flesh-eating bacteria or toxic algae that would go through and disrupt and destroy entire ecosystems, be it in the water or in the tropics, or something on par with that, where we have some philanthropic crazy psycho that's going to say, okay, well, just as you said, we're going to do something to fix plastics, and so we're going to release this microbe that reproduces by consuming plastics, and its byproduct is
So
whatever, sand and oxygen or whatever. But it could mutate, and pretty soon maybe it goes after, crustaceans, or
exactly
it starts attaching to fish or
exactly,
or to something else, plants for that matter. And all of a sudden you've got some biological microbe that's disruptive to a core component within an ecosystem that then creates the problem of, if you destroy the microbe, you're then also contributing to the destruction of a key component of the ecosystem. So you create a chain reaction that you can't stop it.
The whole thing of it is that by natural processes, mutations do happen, but they happen slowly enough that the whole planet has a chance to adapt to it and modify and cope and change. What we're doing now is we're able to do these things. We're able to literally design new life forms very, very fast. That is something that that's this point number one, doesn't necessarily give the Earth, the rest of the biosphere, enough time to adapt, number one. And I forgot what was number two, but anyway, it'll come back to me. I know what it was. It seems like, and we know that this is true. People come along, they fix one problem and create two more. We fix one problem and create two more. It's like the level of complexity of things is just increasing logarithmically, and I don't know where it ends. And anyway, I take this lady to be a very, very intelligent, very well-studied scientist. And the thing that I would like us to think about is, it seems like it's necessary to see the big picture of life, the entire biosphere, the entirety of time from as early as we can track it up to the present. And it seems like people have a tendency to get into a kind of a tunnel vision area where they're saying, no, we've got this thing and we're going to focus on it. We're going to work on that. But then things that are to the periphery that we didn't know could become impactful in the future, just as you said, then come along. And now we have problem number 2 and 3 and 4. So all right, let's listen to her some more with all this in mind.
It basically is rivaling the rate of disappearance when we had basically that big asteroid land on the surface of the planet and basically destroy the dinosaurs and allow mammals to flourish. So this is a huge, I think, rallying cry for, I think, scientists across many disciplines, but including synthetic biologists, to think about how we can really address global scale challenges using synthetic biology. So I'm going to dive down and talk about one very specific problem. So by 2050, we think there are going to be, as Pam said, 10 billion people on the planet. And if we look at what they need, we are going to need not only new energy, so 50% more energy, 100% more energy, we're going to need more food. We're also going to need new materials. So we estimate that we're going to need somewhere between 100 to 100% more materials than we currently have. Yet, because of climate change and because we have already extracted many of the very cheap to extract ores from the face of the planet, we are going to need to do that to create these materials with less energy, with less water, and with basically more dilute precursors. So that is kind of a serious scientific challenge. And what I would say to you is that I actually am inspired to kind of turn to biology as one of the potential solutions for this problem. So biological systems are really amazing in their ability to make materials that have many of the key features we're looking at for. So these materials, so skeleton, bones, teeth, eggshells, the spicules of a sea urchin, these are materials that are grown under ambient conditions, oftentimes with very low energy inputs. They are they have multifunctionality. So they serve many different.
So interrupting, you see the case that she's making is we have some very crude processes for manufacturing a lot of what we need on this planet, and for day-to-day life products that we use, things that are incorporated into those products. And she and many other scientists are seeing the opportunity to program microorganisms to manufacture some of the components that we could use. They might be food, they might be other things, they might literally be structural components that we could use in things that we want to have, whatever they might be. Instead of waiting 100 years for a tree to grow, we can grow wood pulp using some form of bacteria and make paper, and do it cheaper and faster than having to wait for trees to grow.
These are the kinds of technologies that are useful if you're going to start terraforming another planet or the moon. You could take a very small sample of some type of a microbe and then grow it once you get there, and it reduces a whole lot of components that you would need to transport.
Yeah. That's would be a really good use for it. Terraforming Mars or the moon, some place that was lifeless, but it has lots of elements, lots of minerals, and unleashing some form of bacteria or microbe on there to turn those minerals into structural things that we could use, and maybe even create atmosphere.
It'd be more sophisticated than that. You would scale it here or somewhere.
Test it here?
Yeah, or maybe, go to an asteroid or something nearby to do this stuff. So if it goes wrong, it's fine. You could deal with it. But what you would do is you would build a system, a machine, if you will, a platform that would then be able to assemble whatever biological sequence you needed to be created on demand, and then you just transport your nucleic acids, your A, C, T, and G, and probably some uric acid as well. But you just transport that and then have the machine build whatever organism in whatever genetic sequence you wanted it to be when it got to wherever it was supposed to go. Right. And that stuff is stable for long periods of time. Dry, wet, doesn't matter. That's the point that we're at, is where these synthetic biologists can model life in a computer, supercomputer, quantum computer, whatever. They can model life, and then they literally can print it out with proverbial ink cartridges that are the base compounds for life.
They have
nucleotides, I think, is what I'm not nucleic acids. It's nucleotides.
Right.
Yeah.
They have within the wheelhouse now the ability to design, and let's just focus on bacteria. Let's not talk about mice or humans or anything right now. It is within the wheelhouse now to be able to construct, program that bacterium to take in one type of material, which let's say is something that we have an abundance of, and to output something that we don't have very much of, including even generating electricity. And that's in the next video, which we might or might not get to. And it's really fascinating. But again, just try to focus in on what the overall perspective is. I come from a little bit more of a perspective of nature is really amazingly beautiful, and it is kind of like a self-balancing, harmonious ecosystem that regulates itself. And again, we come along in evolutionary terms in a very, very brief period of time, a tiny, tiny snapshot of the time of the history of life on this planet. And we are now ready to make mega changes, mega impact on what types of life are out there. And so what I'm looking for is in the enthusiasm of Caroline Ajo Franklin, that there is that overall perspective. And what I keep looking for is some sort of a first do no harm, or reverence for life, or be careful, or something along that line. And what I get is that she really does see some of these problems, and I agree with her. She sees some of these issues, and many of them are man-made, man-caused issues. And she is seeing the opportunity with the knowledge that she has in her background in biology, physiology, biochemistry, to be able to utilize, to capitalize, to leverage the capability to reprogram bacteria and maybe some other small life forms to fix some of these problems. It could be good, but I see a lot of things that could go wrong. Let's listen to her some more. I could talk all night, and let's see what else she has to say here. I may skip forward a little bit too.
Processes at once. So the eggshell not only is a protection and mechanical physical structure, it is also a structure that has acts as a very selective filter that only allows certain gases in and out, and that's what allows eggs to give life to new organisms. So looking at nature, we can see that there's a huge opportunity for making materials in a sustainable way that could really affect the challenges we face. So that's one of the real things that I think inspires me in this area is the question of can we create a new era of materials and a new era in which we make materials? So we all know about how the invention of the silicon microchip changed how we live. It has given rise to the digital information age. What if we could make a new brand of materials using biology that would be the next revolution and that would pave a path toward a more sustainable future? So right now we think of the natural world as containing things like us, organisms that grow and divide and self-heal. And these systems have these amazing features, but I would say by and large, they're not yet engineerable in the way that certainly CMOS technology is engineerable. Or we have devices. So think of your cell phone, think of your computer. These are highly engineered. We can engineer them to our specifications. They're amazing in terms of the ability to do rapid data processing, rapid communication, massive data storage. But if I throw any one of your phones on the ground and stomp on it, that's not going to fix itself in a couple days, no matter how many Band-Aids I put on it. So could we really move to a future in which we grow devices, in which there is a seamless communication and...
Okay, growing devices. All right. Well, in some form, we do that in a much more primitive way with silicon and with with the photographic processes to make semiconductors, and it's really just taking that same concept and using biomolecules to do it, and that'll make them more miniaturized and probably ultimately more efficient in energy usage. But also, it can be done in a way that you design those living creatures to build their own structures. It's all in the programming. It's all in the software, that genetic code for these life forms. So it is absolutely fascinating. And I kind of don't know where it's going to lead. Let's listen a little bit more. Let's see where else she goes with this thing.
And feedback between biology and materials. And if we could do that, we could maybe make a whole host of new technologies. And Pam talked about some of them.
Autonomous biosensors, okay? Self-powered biorefineries, self-repairing solar cells. Positive ideas, I guess.
Them. For example, the ability to take essentially solar cells that self-repair and create, in a very distributed way, chemicals. So this is what really propels us forward in the scientific vision that we see. So that's a huge challenge.
I'm sorry, I have to stop again. Scientific vision. I'm looking out in the chat right now. Scientific vision is, okay, when we have scientific vision, don't we need to have a very broad scientific vision, a large worldview? You know what I'm saying? Is our view large enough? Or do we focus in on one thing? And of course, we do focus in on one thing to really master it and become expert and precise with it. but a combination of both is, and that big view is what's important for the safety and well-being of everybody, including every other life form on the planet too. So let's see what she got up here. Design rules. Okay. Interface, abiotic and biotic. Okay, well, abiotic would be your electronics. Biotic would be the life forms, whether they be bacteria or cells that become incorporated into things like humans. Transfer information and energy. Okay, don't know where she's going with that. Let's listen to her a little bit more. Integrating into device. All right, let's see what she's got to say here.
Tell you about a couple things that my group is doing to kind of address that huge technical challenge. So one of the things we're doing is we're very interested in understanding at the molecular level how living biomolecules can interface with inorganic materials. There are precedents for this happening in nature, and we really want to understand how we can steal from nature to make new devices. We are also keenly interested in how you can actually use synthetic biology to allow living systems to seamlessly communicate and power devices. We want to make those devices, and this is really much like we've heard from sort of the neurobiology.
Okay, so it's the old argument. Before someone invented the automobile, everybody was riding around on horses, and many people would have just said, I don't need an automobile. I got a horse. I can go everywhere I need to go. So old tech versus new tech, there's always resistance. There's always early adopters. There are always people who are retrograde in their adoption of it. And there are always arguments on both sides of that, okay? So we see where she's starting to go with this. It's efficient manufacture of materials that we may need to sustain a larger population on the planet. But the other thing she's really featuring is that bioneural interface, which we have spoken about on these shows from so many different angles. So she's also got that piece in there too.
She's talking about cyborging nature.
Exactly. Yeah, exactly.
It's beyond just the brain. She's literally talking about fusing the digital,
yep,
the inanimate, if you will, the lifeless with life.
Yep, that's right.
That's what she's showing you there.
Yep, that's in the upper right. Yep,
it's the top line. It's both of them.
But actually, it is both top ones. Yep.
Yeah, the abiotic and the biotic, where she's, you know.
So things are going in this direction. They're going this way, and you know, as we've said before, there are very, very well-financed research labs with very, very gifted people of an engineering mindset who work on this stuff, and we always come back to that same question. Let's see where she's going, and this is one of many people that we've featured, but you haven't heard this lady before, at least not on our show. Let's see where she goes.
Brain interface, this is also a key question of how do you do this integration in a way that makes sense? And the last piece is how we actually use biology to hierarchically assemble materials. So today what I'm going to go ahead and focus on is actually these two pieces of how do we use synthetic biology to move energy and information.
You heard that last piece that she said is how do we use biology to assemble all these pieces, right?
Right.
She said that very fast, but
they've reached a physics-based restriction with the transistor at this point.
Right.
5 nanometers, 5 nanometers, as small as they can make a transistor now for the new computer processor chips. You can make them smaller if you are able to do it with biology.
And if you want to get some sort of input-output into the nucleus of a cell, for example, some sort of an interface, you're going to be well served to do it utilizing biomolecules rather than chunks of silicon.
There's benefits to both, and the further down the road we go, you're right. It's going to end up being exclusively advantageous to use biology, but they can already do that now. They can interface inside of a cell. They can with nanotechnology that's not made of organic material. It's basically silicon-based, metal-based
We can have, they're also, other people are also working on, let's say, biomolecules, but in vitro for the purpose of making logic gates and computational components. So talking about organic computers, in other words, computers made out of organic biomolecules, but outside of a living organism, just because they're really small, they're really efficient, and if you can write the code to build these things, or even better yet, write the code to make them build themselves, then some of those will be outside the human and some of them will be inside the human.
That's the benefit to the organic, is that once you program the organism, as it goes through its life cycle, it's going to reproduce that programming. You don't have to add materials anymore.
Yep. There's a question out in the chat trying to wrap my brain around how you grow devices. Well, it is a matter of defining what are the electrical characteristics desired for let's say, a given logic gate.
You see the
did you
see the document I sent over to you in Zoom?
I don't know, but I can go look for it.
It might help put some context around that question.
Yep.
It's one example.
Let me go retrieve that. Please stand by.
And there's another document I'll pull it up in the background to explain what a nano network is, where it's at now, and where we're going with nano networks as they were projecting it back in 2012.
Which one were you speaking of that you sent me?
So I sent it to you in chat in Zoom.
Oh, I'm looking in the wrong place. Please forgive me. I can get right on that. Oh, there we go.
Mm-hmm.
Loading.
I guess I could have just shown it. I'm gonna dig the other document up real quick.
Okay, I'm bringing this on board. Oh, it's a PDF you sent me. Yep.
Yeah.
Pretty good size too.
It's a few pages. Yeah.
Okay. Half downloaded. Okay, so anyway, back to while that's downloading, we literally grow silicon semiconducting devices in the world of silver, gold, silicon, and photographic processes by which we project the image of the circuit that we want to make onto a template and then develop it and then connect wires to it so that we can use it. It's really the same kind of thing, except substituting biomolecules for it. So now let me open this file.
Conceptually, it's the same. I've got the share. I took share.
Oh, there you go.
So towards molecular computers that operate in a biological environment. This is back in 2008. And this was out of Israel. And what this is showing you is the use of A, T, G, and C as logic. Based on genetic sequence, it will do something or it will not do something. And you can start creating pairs and partnerships in order to create what they call Boolean logic, Boolean algebra.
Yep.
And so that picture isn't completely comprehensive, but what you're looking at here in this case.
That's software. That's RNA. Software.
Right. And if the RNA lines up properly, then it will perform some function. And this is where you got to get back into a little bit about how RNA transcribes information, because if the right environment isn't there in the cell, then RNA won't complete its process.
Right.
And it's part of the logic piece. You have to know all of those if-thens about it.
Right. That upper left is showing a sequence that won't proceed. And then the section B looks like it's showing a sequence that will proceed, I think.
Yeah, I'm going to read this caption here in figure 3 to give you an idea. This is, like I said, this is just one example. You have entire institutions working in this field, but it says, in this case, controlled release of an active drug, a short single-stranded DNA molecule upon a positive diagnosis. So they can introduce medication into the body and it won't release the medication the delivery vehicle won't release the medication unless the right signature is there to interact properly with the RNA or the genetic information that is wrapping up and enveloping the medication.
To liberate the medication for general release into the body.
It'd be like a lock and a key. I kind of misspoke that, but you get the idea.
Absolutely.
So it says 2 types of molecules that share the same diagnostic moiety, but differ in their output moiety were exploited. And what they're saying is that you've got a few things in here that are very, very similar, but they've made a delivery vehicle that can distinguish between what's different about these similar items. Each of these output moieties have a stem loop structure. The stem, which is a light colored, holds the functional part. The dark color is inactive. So up here, you've got the yellows and the grays, right? These are the parts that are going to basically decide yes or no. And the darker color is going to be delivering whatever it is, genetic payload, medication, whatever it is.
And what we're talking about basically is just a machine. Humans have made thousands and thousands of machines. This is just a machine at the size and scale of genetic molecules. It's just using some different stuff to do what a machine does. That's all it is.
So before we move forward, that's a really good question. And it's important to frame this, especially if we're going to go a few episodes.
Yeah, we gotta. Yeah, it's going to be very, very interesting.
So nanotech, right? This is where we're at now. This was in 2012 is what you see down here. It's just like the year that the slide was created or distributed, whatever you want to call it. And up here it says 2010, journal, March 2010, Nano Network Journal. And you've got a self-powered nano chip that is
Look how small that is.
Right. 6 micrometers by 2 micrometers by 1 micron. You have a nano processor, nano electromagnetic transceiver, which is a antenna, right? Nano antenna, nano memory, a nano actuator, and nano sensors, which can go in and do things like sense biomarkers, changes in pH.
That's only about the size of a virus. Yeah. That's really, really small.
Yeah. Like you can inhale this stuff and wouldn't even know it.
Yes.
This could pass through a needle and you wouldn't even know it. This would pass through a vaccine needle and you wouldn't even know it.
Yep. That's right. Robert, good to see you out there.
So this is old tech at this point. We've been living in this world for a decade or more.
A decade. That's right. More. You guys out there in the chat, fire away your questions and comments. We're watching.
So nano sensors, to give you an idea of what this, nano sensors up here, they kind of break it down. We're going to go and take a couple minutes and go through this, Indy.
Yep.
Force sensors, chemical sensors, biosensors, biological sensors. So look, you have antibodies and antigens. You can dope them and put it on some nanotech and put it in the body, and it acts as a sensor to understand if you've got particular resistance, antibodies to something, or if you're immune to something, or if you're
or whatever your chemical environment in your body is, sensing your biochemistry, your temperature, your pH,
anything.
So, adrenaline level, whatever it might be.
Yeah. Nano power generators, graphene for solar cells. So they're kind of going off into commercial applications here.
Right.
But nano power generation is important because that's how you self-power this stuff without tying it to a power supply.
Right, which is very handy.
Oxide nanowires, vibrational energy harvesting. So they can harvest energy with zinc oxide nanowires just by these wires moving. Piezoelectric, the conversion of mechanical movement into electricity.
Yes.
With nanotech. Nanoprocessors. So IBM, Qualcomm, Samsung. Back then, right, 2012-ish, they're already at 32 nanometers or 20 nanometers. They're down to 5 nanometers at this point. World's smallest transistor in 2008 based on graphene nanoribbon is just 1 atom by 10 atoms, a 1 nanometer transistor. Back in 2008, they're saying they were able to successfully make a 1 nanometer transistor that's 1 atom by 10 atoms, 1 atom high, 10 atoms long.
But that's graphene. That is not getting into the territory of using biological molecules. That's graphene. So now, when you make this stuff out of adenine, cytosine, guanine, and thymine, which are the components that make up RNA and DNA, now you can go down to an even smaller size and get more circuit density in a smaller space, and arguably definitely more efficient as well.
So you have a hybriding that was done. It's already been done. A hybriding of DNA being used as building blocks to be able to steer and direct and precision assemble and precision target and precision locate where this synthetic nanotechnology can go. And so it was like the glue and the popsicle sticks that make up, the proverbial Tinker Toy set kind of thing, right?
Yep.
And I'm saying all these things as cliches just to give you an idea. These are construction materials. They're using biology for that back then and still are. And then we move into nano antennas, graphene plasmonics, which beyond the scope of the discussion today, terahertz waves, because those are small enough to interact at that scale. We're getting into the synthetic biology here real quick.
They've got all the studies that characterize how this, what its physical and functional characteristics are.
Yeah.
And this
we're getting there. This is where it's going. This is where it is, and this is where the bio side's going as well. Nano networks everywhere. Nano networks out of your brain, nano networks throughout your body, in particular organs, and enabling full connectivity throughout your body at the cellular level. This is what this picture is showing you.
That's a great diagram. This is 10 years. This was in conception 10 years ago.
AT&T had a patent in the same year to do this very thing, to network down to the cellular level. So we're way beyond this at this point. We're way beyond inserting synthetic into the body, which is why we're going over the synthetic biology, because where this is going is to rewrite the genetic code of the planet so that the planet becomes the Internet of Things. It's not just going to be walls and windows and cars. It's going to be trees and flowers and bees and bugs and you. And it's going to be grown in you through your life cycle as a result of genetic modification. But we have to understand where we were and where we are to be able to visualize where we're going, and why statements that the lady made earlier about us doing something that might disrupt the life cycle on the planet being a very real concern. Because that's what we're doing at this point, is we're manipulating the literal software code of life on this planet at this point.
My thing, TMC, is we're doing it very, very fast. So it's not going to allow 1,000 years of time to see how the natural biosphere reacts to it. We're not going to have that. This stuff is going to get
they're modeling out. The best they're going to get is modeling out of supercomputers or quantum computers to model what ifs, run scenarios that would take hundreds of thousands or millions of years to realize or conceptualize with classical computing, or just to allow it to occur. They can model it.
Yeah, but how many flawed models have we seen? Okay, data in versus data out.
When's the last time you had an accurate weather report?
Well, they vary,
Yeah.
So there's not enough consistency there, in my view, to merit getting. I mean, it's literally tinkering with the genetic code that nature has balanced and evolved over many, 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 many years, and it's done so in a way that it gave it enough time to correct itself if something went wrong.
Yeah,
we aren't going to have that kind of time.
So nano cameras, other nano things, nano projectors, ultrasonic nano phones, right? This is when you think of all that stuff, you're like, okay, well, that's manufacturing plant. That's like, stuff that would be made in China, or, the next generation of a computer or phone. what I see when I look at something like this is these are the synthetic sensors that make up the network of the Internet of Things that are going to be transformed into the sensors that are going to be directly reading information out of your biology.
Question from the chat is this one-way or two-way? It will almost certainly be two-way, bidirectional communications. Yeah. Absolutely, almost certainly.
Yeah. So we're getting into, it's a great question. We're getting into bio-inspired design. So here it is. This was from was it Virginia Tech or Georgia Tech? But this particular presentation was passed around a few of the leading schools. Silicon technology era is coming to an end, 2020 to 2030. That's right now. Molecular technology era is starting and will be dominating our lives for the next 80 years, around 2020 and beyond.
And we're there
now.
This is true. Yeah. This is it. The prediction.
So biology.
Yep.
Radically different approach to nano machines, and we were looking at electrically connecting cells. It's beyond that. It requires nano machinery to do what it is that we set out to show today. And
those are things that will self-construct inside of an organism.
Right. Or to grow.
Onboard instructions to build themselves. It's like they to these devices, they look at the organism that they come to inhabit as blank real estate, raw land. And they say, okay, I got energy, I got proteins, I've got electricity, I've got vibrational energy to power myself, and now I'm going to start building some stuff in here. And it's all going to happen because they have onboard instructions, that is software, in them at nanoscale that will allow them to start building whatever devices, literally organic devices, inside of the organism. Well, and that is essentially them looking at that as a rich territory of natural resources that they can use to build themselves, whatever it is that they're going to build.
Yeah, you'd have to genetically modify these organisms that are designed to just reproduce. And so the goal is to, if you're going to produce like synthetic, I think the example used earlier was wood, or synthetic materials for construction.
Yep.
You would use something like yeast or bacteria to mass produce some chemical or some component, some byproduct or something you can harvest out of those large bacteria population, just to use a term that's on the screen here.
Right.
And that's the real value of the simple reproducing type of organisms, is that if you recode them to produce or excrete or mutate into something that's useful for humans, then all of a sudden now you've got a very easy way to create something without a whole lot of harvesting. I mean, it's done on demand.
In a way, it's very elegant. It is very efficient. There can be a lot of good things that can come out of this. The only thing that makes me a little bit nervous is engineering these life forms, and mutations do occur, and the unexpected does happen. And if you made a great example before, we don't need to go over it again, but an unexpected, design one of these things, and unexpected results, such as, okay, it starts feeding on crustacea as opposed to plastic.
Well, here's a great example. They make something that biodegrades plastic, but over the last 20 years, we've got this whole recycling fever where, well, this stuff's made with 30% biorenewables, and this is made with 30-40% plant, and it biodegrades, so it's not as harmful. Well, if you start making things that break down a plastic bottle and it's not efficient enough because you've got so much in circulation that's made of organic material that you have to tweak your recipe to get it to market, to sale, all of a sudden now you've got something that very easily could mutate and start chewing up grass or trees or leaves or whatever, because it's biomass that's been put into plastic materials under the guise of saving the environment.
Right. That's a great little diagram that you have up there right now.
Yeah, I thought you liked this one, bio guy. So this is a biological nanomachine, a cell, as a diagram to illustrate beyond just a communications perspective, but it's actual machinery. This is a miniature factory.
That's what cells are anyway, TMC.
Yes.
They are all little miniature factories, and we've got quadrillions of them in our bodies, but they are operating under natural instruction sets. They're operating under what has developed, what we've been given through our genetic heritage. This is changing that and changing these little factories to do something else. And who is going to be doing the programming?
Yeah. So the reason I think we should go through these, or at least leave them up for a minute, is you're blending. What you're blending, or what I should say humans are blending at this point, is computer science, data analytics, biology, and networking technology, amongst other things that are just not coming to mind at this moment. And they're fusing it all together to rewrite, interpret, and redesign life. And you gotta understand what's going on in chemistry, what's going on in biology, to be able to make those translations from silicon, right, computer stuff that we understand as computer, and then transform it into biological stuff.
Well, their motivation is to move from silicon into organic molecules because they want to keep pushing forward with the computerization and the Internet of everything. That's one thing. But the other thought that I have is nature does build things. It makes things too, but it does it in a different time scale. I'm all about the time scale here, and that's my thing.
Yeah. So here's the nucleus of it all, no pun intended. It's getting into the genetic code in order to instruct the entire cell, the organism, to do something that you want it to do. And so DNA, right, contains the information to basically create the protein structures, memory, what they equate to memory in a computer circuit, computer chip. Ribosomes read and process the DNA. They're the processor. They synthesize the proteins. And the proteins control the functions of the cell signaling and the receiving of signals from other cells or other things that are going on within the same cell. All of those things are necessary for a factory needs communication. A factory has to have order. There has to be an order of operations, or you don't get a nice set of Crayola markers when you're done. The red might have a green insert in it, and it might have a purple label on it if the order of operations is not correct. And that's why understanding these components in a cell or in life is so important, because they all have the right ordering mechanisms needed for it to function as intended.
The question that comes to mind is, with this level of understanding of the inner machinery of cells, when we have a person who has diabetes and their pancreatic cells do not function right, or they're not getting their signaling quite right, it would seem like we have well in hand the kind of technology to do minor programming changes, or to do minor reconstructive instruction to things like an organ like the pancreas, or a kidney for that matter. And we could be paying attention to that and helping to cure diseases. Cancerous processes are generally cells that have gone out of control and they're multiplying uncontrollably. And we could fix a lot of things by doing conservative repair work rather than, this looks like pretty major stuff here.
Well, these are the basic building blocks. And it's going to be true for genetic therapies as much as it's going to be true for, designing yeast to excrete or express magnetic properties. Yep. It's the same underlying platforms or principles, you have to understand what you're doing with the DNA and the RNA, and you have to be able to read and understand what the ribosomes are doing with that new code, and then what proteins are being made and how those are interacting in the cell. Otherwise, you get unintended consequences. And so you're right. What are we doing? But the real underlying, I think, point here is that these are the fundamentals to understand what you're doing with biological life at the cellular level. It's different than biological life at our level that we generally understand, where I eat this kind of food and I'm going to, I'll get skinnier, I'll get fatter, I'll stay the same. I work out or I don't, and I'm soft or I'm not, or I'm fat or I'm skinny or I'm fit or I'm not. I don't sleep enough. I sleep too much, right? Those all impact a bunch of stuff that goes on with your biology. But do you have a specific understanding of what it's doing to your DNA day to day, different parts of your body and the DNA within those subsets? Do you know what a lack of sleep is doing to the DNA in your brain versus in your biceps and your triceps?
Mm-hmm.
We don't. And this is the level of specification and detail to do what it is that we're going to show over the next few episodes here. You've got to get down to the proverbial nuts and bolts of what this does. And so this document is oriented around cellular communication, and it loosely ties in with what the lady's talking about, electrically connecting cells or whatever it is. But just understand that cells communicate with each other in colonies. Your body is full of cells and they all communicate, and there's many different ways that that's done.
That's right.
And so you've got the movement of chemicals and proteins and ions and in this case they say hormones, which are technically chemicals, but they all do something. They all signal something. They all are excreted as a result of some action that's occurring inside or outside of a cell. And these communications vary in their intensity, and they vary in their speed, and they vary in their reproducibility, meaning that once certain things occur, it might take a moments or minutes for it to be able to reoccur again, especially when you're dealing with like chemical signaling. It's a very, very slow process in it, and it takes time to reset because the chemicals have to diffuse again before you're primed to be able to receive a signal. And this happens inside and outside of cells, and I'm not going to really get too far into it, but just
right within and between cells. Yeah.
Right. So
we live on a planet of cells. They're everywhere. They're in everything from a jellyfish to a mouse to an ant to us. And they all work in some sort of a concert. All in any given organism, the cells are kind of like an orchestra, all, playing the song of its life, whatever that creature's life is programmed to be by its natural DNA code. So here we have the fantastic frontier of us going in and programming that ourselves, programming life.
I'd like to show you what they're implying here, what they would like to do. And by using DNA and RNA, what they're trying to do is blend these ideas of putting sensors on something as small as like a flagellum, infrared sensor, temperature sensor, touch sensor, humidity sensor, and a flagellum motor. So
it actually is an organic motor, by the way. The
right.
Proteins that make up the thing that spins a flagellum around are a biological molecular motor. Yes, they are. It actually spins.
We're in the world of completely reprogramming life and or making a hybridized bio machine, a cyborg, if you will. And that's pretty much it for this document. I guess we can, here, communications real quick, different ways that these things can communicate.
We're doing great research, and we're doing some really, really dangerous tinkering, and it's not going to stop. They're going to keep doing it.
Yeah, it's going to keep going. Yeah, the rest of this is a communication oriented diagram. If you want to look at this yourself, go to Human Internet on Twitter, and you'll see this thread here. Where is it? Yeah, it's in a pinned tweet. It's at the top. If you go to Human Internet, it's Human Internet 1 on Twitter, and then it's the pinned tweet. And if you go into the pinned tweet, you can go down and you'll see what looks like this document right here. And it'll take you through the networking components and nano networks, what they are, where we're going, all of that. There's other stuff in here too, and nano-sized RFIDs and Huawei, human bond communication, and some other stuff like AT&T's patent to allow one person's brain to control another person's body. They actually patented that. At the cellular level, they can network your body and your brain at the cellular level, assign subnets to the different centers in your brain and the different regions of your body, like a hand or a foot or an arm, all the way down to a single cell. Network it, and then be able to assign instructions to go to and fro through your body that ultimately come from somebody else's brain. That's where we're going. And this was originally patented in 2012 and then revised in 2015. Anyway, I'll drop share if you want to go back to that nice lady.
That's fine. That fit right into the whole picture. There is a big picture. All right, we can go back to that. And I got another guy I want to get to also. So here we are.
And how do we integrate this into devices? So one of the key challenges that really brought me into this field was...
So you know what? We've heard most of where she's coming from. I'm going to go to another guy now. All right? And this is Derek Lovely. And they're once again, brilliant people. Derek Lovely has done a presentation once again 2 years ago, protein nanowires for sensing, energy harvesting, and wiring cells to electronics. So he's going to use the term wires. He said, well, we got wires. When he's speaking of wires, he's not speaking of copper or, something metallic. He is speaking of molecular wires made of biomolecules. So every time he uses that word, think of these wires being something that are made out of the building blocks that are in our bodies that can be instructed to form into certain structures, creating wires. So the idea is that once you've got enough wires and they're organized in enough ways, even though they're organic biological wires, now you've got a circuit. And once you've got a circuit, now you can start doing some sort of input-output out of it. And he's talking about building these things out of that stuff. So, and he will get into it very, very technical, but we're not going to go that far because it's not necessary. We're trying to get the picture of what this stuff can do and what the end, what the goal is that they're shooting for. So this is Derek Lovely, the DeLange conference, and it was in 2018. Here he goes.
It's a real pleasure to be introduced by one of the early proponents of nanotechnology, Pam and Caroline, introducing sustainability in electromicrobiology. I was wondering yesterday how we were going to make this transition, and I really appreciate them going ahead and making that. And what I'd like to talk about is a revolution.
Right away, the nanowires are made of protein, all right? They're not microelectrodes made out of gold or silver or some other metal. All right, here he goes.
nano-electronic material that we discovered in doing some just basic microbiology in various sedimentary environments. And I realized this is an interdisciplinary audience, so I summarized my whole talk on this first slide because I realized I had to put some data slides in to try to impress my microbiology colleagues. Some of you are going to zone out, so this is, you can get it all right here. I'm going to talk about the microorganisms that make these protein nanowires. Some of their, and quickly, their biological role. One is, for example, transferring electrons to other microbial species. But then when we shear these wires off the microorganisms, they are a really interesting nano-electronic material with ohmic conduction properties.
So sharing wires from one cell to another. So one bacterium to another might have a series of wires made of protein going between them.
Using these nanowires in wearable electronic sensors. I'll talk about some other applications. I have a colleague making injectable brain implants with these. And also, given the discussion.
Did you catch that? Injectable brain implants.
Yeah, and the next step is to understand the genetic profile to make these things and express these things and then tell the cells to connect to each other. All of that requires genetic instruction.
Exactly.
And rather than inserting these already manufactured nanowires, they would just go in and reprogram the brain to grow them upon their own, using the brain's own energy sources, using the brain's own normal cellular respiration, cellular reproduction processes.
We talk about software, and people who are not computer geeks, when you say the word software or code, then maybe their eyes glaze over because that's just not their thing, But we've got code upon software code built into us by nature. And these guys are working with code as well. And he's talking about coding so that you're making proteins that turn into conductive structures for making a microscopic network of interconnection inside of anything. And that this is their work. And we won't go this far, but he talks about how they've refined it. So they've made it so that the amount of advantageous conductiveness of these proteins that they've been building and trying to build has gotten better. So their first protein wires that they were making for incorporation into a living being were not really that conductive. So then they worked on it and they worked on it and they increased the conductivity of their protein wires by 1,000-fold from where they had started out. So they've made great progress with this. And so just, keep in mind, like, where's this going? Once they're able to build wiring and it has good conductivity and we can make connections, and you take a look at the rest of this, wiring cells to electronics. This is what these guys are working on, wiring cells like your cells or my cells to electronics. This is Derek Lovely. So here, we'll go a little further here.
About energy generation, I'd like to talk about how these nanowires can be used to electricity literally out of thin air. So, okay, one more. I'm pushing up, no wonder. We'll be here for a while if I keep doing that. Okay, so we first found these protein nanowires in this microorganism, Geobacter. It's a common constituent of soils and sediments. This is a transmission electron micrograph. This is Geobacter colored in red. And hopefully you can see against this blue background all the white filaments emanating from the cells. Those are the protein nanowires. When they're still connected to the cells, we typically refer to them as electrically conductive pili or E.
So, protein nanowires, here they are. And what these people in their lab are going to attempt to do is to control how these protein wires are made and what they connect to. And that is a slide from an electron microscope.
pili for short. You can see that they're very thin, they're only 3 nanometers in diameter, but they can range from 10 to 30 microns in length. So they, since they're electrically conductive, they greatly extend the electronic reach of Geobacter.
Now think about that. We don't usually think about the idea that a bacteria, which is, I mean, you'd need a microscope to see it, has a whole bunch of little electrodes coming out of it naturally, and it connects to the environment around it. We don't usually think of that. Well, I didn't when I had microbiology 40 years ago. We weren't thinking about that at all. But what an amazing world that we live in, because bacteria are relatively primitive cells. So I wouldn't think it to be too much of a stretch to think that more advanced cells, prokaryotic cells are things like bacteria, primitive. Eukaryotic cells are more advanced, things like the cells that are in humans. And I wouldn't think that it would be very much of a stretch at all to consider that human cells have also got these projecting nanowires all around them that allow them to communicate to the others.
And one of the reasons that Geobacter needs a long-range electronic reach is that it transfers electrons to insoluble electron acceptors. So we first found Geobacter in sediments of the Potomac River. Based on this property, it can oxidize organic compounds to carbon dioxide and transfer electrons onto iron oxides. Well, iron oxides are insoluble minerals. So the big question was, how can Geobacter get electrons that are generated from central metabolism inside the cell, outside to this cell, onto this insoluble electron acceptor?
And that might be enough of that. This, you kind of get the idea what he's working on. Isn't it utterly fascinating stuff?
He's working on another angle to what that other lady, I forgot her name, what she was working on, trying to blend the organic with the inorganic in such a way that it's more or less biostable.
Yep. Right.
It's got to be I mean, he's talking about growing these things and having a colleague injecting them for bio or brain neural implants, right? So that's the goal. Right. The real goal is to figure out how to blend the inorganic with the organic, the alive and the dead, and ultimately to be able to connect life to the internet.
Yep. Pure and simple. That's the general thrust from so many different labs, is connecting life. And I mean, don't think twice about it. It's connecting human life to the internet, to computers.
It's one of the major pushes.
Intelligence. It's a huge push.
Well, here's the benefit to it. If you look at how you define critical infrastructure, communications is a cornerstone. Transportation, communication, and power, right?
Yep. Gotta
so what they're doing with synthetic biology is they're trying to figure out how to use it in modern life, how it can replace things in order to bolster resource management, so to speak. But the reality of it is it won't ever really be efficient or able to be scaled to a autonomous kind of a manufacturing level scale without the communications piece. And so you need that piece in order for this stuff to scale and be sustainable and to be able to control it.
I got to take us out to the big picture. Great comment out there. We are given, people are given, in this lady's view, God gives people the gift of a brain to create. And people take it to destroy or change those creations. And without focusing in on any particular person's religion, there should be a kind of a reverence, almost a kind of an amazement at the beauty and wonder and flexibility and complexity of life. And that I continually come back, whether we're talking George Church or anybody else, just come back and say, these people are thinking that they are, I don't know, they're just appointed to be able to go in and change and monkey around with everything. And I don't feel that reverence I feel that there are people who are very smart and they just move right forward. And I don't know that they ever stop to think about whether they should or not. This is a question we've come back to it in the past, and we're back on it again. And so Derek Lovely is here. Once again, a really, really smart guy, and from UMass Amherst, to my understanding here. And he is just pushing right forward on how to exploit the capacity of these protein nanowires for those connections. And I just have to ask myself, why do I need to have myself wired to the internet? Why do I need to be wired, even if it's protein wires? Why do I need to be connected to computers direct, a direct line from my body? I just don't get it. Is it me? I don't know. There's some obvious fun advantages, right? But don't we have other ways of accomplishing a whole lot of those fun things without going to this extreme? And it seems extreme to me, with much danger. He goes on.
And it turns out it's these electrically conductive protein wires. Geobacter specifically expresses the wires when it's presented with iron oxide as an electron acceptor, and we found that electrons can move through those wires onto iron oxides, which tend to associate with the pili. But it took us a long time to actually document the conductivity or the actual, we knew they were conductive, but the actual conductivity of individual wires. This took the development of electrode arrays. And we can now measure this conductivity under physiologically relevant conditions. And you see an ohmic-like response, and this conductivity is more.
So he's doing some preliminary research on how to really start manipulating these things. And he's come across something really, really good, which is these structures convey electrons. And when you can control the conveyance of electrons, you can make electricity. So I'm not going to play the rest of the video. It's 40 minutes long. I want to move to something else. But some of the takeaways from this are, yes, creating a protein nanowire implant in an organism to be able to connect it to the outside, but also using perhaps single-celled animals, bacteria, and the like for energy harvesting, for literally generating electricity. Very interesting stuff. So I'm going to move on to this one. A little bit of a change of pace here. This is what we started off on at the beginning. And let me just get it sized so it's halfway readable. And I go up to the top. Okay. Redesigning life. So, the promise of synthetic biology. Well. A whole new world. I'm not sure the significance of 1843, so I'm going to go right beyond it. And we're going to get to the article. All right. So, biology is and has always been a way of structuring matter at a molecular scale by putting each atom into its needful place. Again, nature has done this. It's done it for a long time. All right? It is a way of controlling flows of energy on every scale, from that of the smallest living cell to that of the whole planet. Once again, Mother Nature. It's been doing this. Let me see if I can zero this in a little better. It might work. It's a way of growing order and surprise in a universe that in all other respects tends towards entropy. Yes, life creates complexity. In the absence of intelligence and in the absence of life, things tend to move toward a greater state of disorder. But when intelligence is present and when life is present, things move in the opposite direction, toward complexity. Now, what else does he talk about here? Yeah, we're going to skip around a little bit. Any thoughts, TMC? I'm going to move on through this a little bit.
Yeah, no, I think we should keep going.
Where to? Where at?
That's fine. Wherever you're going to go through. I don't know if you want to go to the history or the where we're going.
Yeah, that's history. That's history. Okay, I'm going to pick up right here.
Yeah.
Let me zero in on this one. As DNA synthesis became more widely available in the early 2000s, various ways it could engineer new capabilities into organisms began to come together. By 2002, engineering undergraduates at MIT were using genes bought online to transform bacteria. In 2003, Kiesling and colleagues founded a company, Amaris, with an eye toward making artemisinin, I don't know what that is, and other useful stuff. The media got wind of the excitement. It was not just that synthetic biology seemed like the sort of thing from which weapons could be made. Scientists playing God is always good copy, even if the creations were, as of yet, only microbes. Rarely has science offered such a heady scent of goddishness, with forbidden fruit undertones of Frankenstein and Faust, and raised so many ethical dilemmas. Well, there is a bit of an ethical dilemma. I think I'm more keyed in on who controls it, who's deploying it, and whether there is some crazy guy or girl out there that designs something that is a kind of a runaway bug that wreaks unexpected havoc on a whole lot of things. There is significant possibility that could happen. Okay. All right. So another, this is kind of an overview. Drew Endy, a charismatic young leader in the MIT group, talked of re-implementing life in a manner of our choosing. George Church of Harvard talked of synthesizing not just genes, but whole genomes, including perhaps those of creatures now extinct. Amid all this revolutionary talk, young companies in the field made the fateful decision to plunge into biofuels. It seemed a noble undertaking. Biofuels could usher in the new technology of life while making good the damage done by the old technology of industrial machines. Governments were keen to subsidize them, but scaling up the pathways that produced hydrocarbons by the gram in the lab to the scale of millions of liters proved to be much more difficult than expected. So the effort at making biofuels didn't really work out too well. Let's continue skipping forward. It's an overall survey here. And this is all, this is written generally in a kind of a non-technical, historical kind of a thing. And it's in the magazine, The Economist.
At the bottom of that, the last paragraph you're reading, I saw the word in there, DARPA. They might have been explaining there who else was interested in this, in this field.
That was back over here?
Yeah, the last paragraph that you just read. I don't know if it was off to the right or wherever.
Okay, got it. Yep. All right, let's continue. The capital expense for the biofuels was huge, and then oil prices fell. So the biofuel project failed. That made investors very cautious about synthetic biology. But the field had attracted a bit of support from some governments, such as those of Britain and Singapore. In America, the Pentagon's Far Out Ideas Department, DARPA, which had taken an early interest, going over to the next page, bear with me, created a new Office of Biology in 2013. DARPA created an Office of Biology.
It's their BTO. It's their BTO office, Biology Technology Office. Yeah, DARPA BTO.
And 2 years later, it launched a program that paid for leading laboratories in the field to put together pathways which could produce thousands of molecules never created biologically before. Okay, so DARPA got involved, and now we're fast tracking. Now, they say, yeah, we want you to make more molecules, make more organisms, grow new stuff, come up with new spliced genetic code. So In the past few years, synthetic biology has shown signs of starting to live up to its promise. In part, this is because of academic effort and its cumulative gains. In part, it is a matter of startup companies in the field finding their feet. But other factors are also involved. One was gene editing technology, which we featured I don't know how many episodes ago. Ways of doctoring the existing genetic code one letter at a time, and that refers to that CAGT. All molecules of life on this earth are made of that same genetic code, just in different sequences. In 2000, there was none, but now there's a whole range. And those based on a molecule called CRISPR have proven particularly powerful and easy to use. This has breathed new life into the idea of making precise changes to genomes, which is what synthetic biology is all about. It has opened new fields for biological research and new investors' wallets to the effort. Nobody saw CRISPR coming, gene editing technology. The falling cost of DNA synthesis, on the other hand, was widely foreseen. But it has been a dramatic enabler. The price of a gene synthesized to order is about 1,000th of what it was at the turn of the millennium. If you buy in bulk or have technology in-house, it can cost even less. And then, this is where this whole picture all starts to kind of come together. And then there is machine learning. Synthetic biology gets even greater benefits than most other industries from the recent growth in the capabilities of pattern recognition programs. It is not just that the laboratories produce reams of data with which to train such programs. In 2005, Dr. Endy noted that the designs of natural biological systems are not optimized. Let me see if I can get this zeroed in a little bit better here. Designs of natural biological systems are not optimized by evolution for the purposes of human understanding. Can you deconstruct what he means by that? Designs of natural biological systems are not optimized by evolution. So humans want to go in there and they want to redesign those natural biological systems to optimize them for current purposes. Okay.
Yeah, I think what he's saying is that the way that biology has evolved is not optimized for us to understand how it evolved. So what he's saying is that there's some nuances there that are difficult to identify, which is why the machine learning and the pattern recognition is so important and is so transformative, because the manner in which the computer can understand patterns would supersede or be better at identifying unique patterns or certain data sets than a human.
Yep. So it's a kind of a coming together of the ability to get into DNA and be able to pull the code out of it, and then take that and plug it into very powerful computers and find patterns and uncover the rules, as I quote him from this article, which is right there. Anyway, this is not science as scientists understand it. But if rigorously tested, these rules can be a basis for engineering. They were good rules for building bridges long before there was a theory of gravity. There were. While synthetic biology has grown more capable, the promise of two older approaches to the improvement of life has diminished. One is the pharmaceutical industry. Seems bound by Eroom's law. That's Moore's law backwards. I never heard that before. The number of new drugs developed per $1 billion of R&D spending falls remorselessly. It was 10 in 1970. It is well under 1 today and still dropping. This excited interest in fundamentally new approaches to medicine. One is Getting away from the chemical pharmaceutical approach is to reprogram cells to do therapeutic things. And once again, this really could be a great territory for helping to unprogram cancerous cells in a person's body, or to reprogram malfunctioning liver cells, or to just plain grow new ones from stem cells right where your liver belongs. All these things are within the realm of possibility. They're coming closer and closer. Immune system cells are the most obvious candidates. The cells of the microbiome, excuse me, the interlinked bacterial ecosystem that thrive on skin and in the gut are another possibility. What do you think? Should we go elsewhere? This is kind of a historical here. Well, I'm going to go over to this section. Here we go. And this is synthetic biology differs from everything that has come before. To understand biology, synthetic or otherwise, you have to understand how proteins are made. Proteins carry out almost all the basic functions of life, from respiration to reproduction. And they are made of 20 smaller molecules strung together into a chain. Those 20 smaller molecules are called amino acids. The shapes that those chains fold up into in order to fulfill different functions are complex and incredibly difficult to predict. But they are almost all entirely determined by the order of these small molecules called amino acids. The gene for any protein is simply the list in order of the amino acids required to make the protein. The information is written down in the genome as a sequence of DNA bases: adenine, cytosine, thymine, and guanine, that is A, C, T, and G. In the same way, data in a computer is stored as a string of ones and zeros. It's really just code, except it's the software of life. The programs that turn these DNA sequences into sequences of amino acids is the genetic code. It assigns amino acid meaning to each of the 64 different three-letter words, such as ACT or CTG. Did you know that there were 64 of them, TMC?
No.
I didn't either.
Yeah.
So this is what it is. So when we talk about what these two scientists were speaking about earlier, when they're talking, well, Derek was talking about making electricity with bacteria and being able to program it in such a way so that those protein wires form in a particular way. And Caroline was talking about things such as, using life as the most efficient factory that we ever had. Because in her view, we need to rescue the planet from whatever, she sees coming down the road, which she suspects is not good. Food shortages, diseases, whatever it was. So anyway, these scientists are using this code book, and they are attempting to figure out how to code genes so that cells will carry out the appropriate beneficial manufacturing process to create the output desired. So the cell carries out the same algorithm, but instead of a code book which matches codons to amino acids, it uses codon recognizing amino acid carrying molecules, and this starts to get technical. So I'm going to skip over that. Basically, they're just talking about how that factory works. And here we have a few diagrams, which you can just look at just for fun. Particular codes. Notice the complementariness of these. A, U, adenine and uracil. A, U, A, U. They always code in the same types of, there's a mating pair. So cytosine and guanine always pair. And constructed of this code is all of the genetics of life, basically, in short. Now, where should we go from here? should we? What do you think?
I don't know. Where does it say this protein design?
Where are
This
you?
is a protein design and DNA synthesis now make it possible to produce proteins that separately or together do things nature does not. They remain imperfect. But because DNA sequences are cheap, it is possible to try lots of variations to see what will actually work. Thus, for example, there are companies working on new metabolic pathways which combine enzymes freshly discovered through the sequencing of ever more genomes, enzymes long understood and enzymes significantly re-engineered. It is an exacting craft or art, and it requires not just finding the right enzymes, but also bringing about the carefully balanced levels of gene expression needed if a dozen or more of them are to work together. I'm just having visions of that mRNA, COVID thingy. Not to mention tweaking the underlying metabolism to prevent things produced by the new pathway from disrupting those already there. But if the work is done well, it seems now to be the case that more or less any small molecule found in nature can be made by yeast or bacteria in a fermentation tank. That's huge. So it seems that's huge. It seems now to be the case that more or less any small molecule found in nature can be made using yeast or bacteria in a fermentation tank.
Right. Just load the construction code into the bacteria or the yeast, and they'll make any biomolecule on the planet to order and in quantity. So then they go into particular examples. Yeah, that's okay. Okay, nature uses 20 amino acids in its proteins, but there are hundreds of others that could be used, some of which would confer interesting new properties. It will be possible to make the tag codon mean one of these other amino acids by designing a new transfer RNA molecule that recognizes the codon and new enzyme to stick an amino acid to that molecule. So, yeah, nature's been, once again, tootling along with pretty much just a group of 20 to make every form of life that you see on the planet. But there are other amino acids that could be used to make heretofore unseen life forms that, they might be viable. They might continue to persist or subsist or grow or maybe multiply. We just really have to hope that, since it doesn't seem like we can stop this, we just really have to hope that people that play with this stuff can control it. Here. What do you got?
Bespoke genetic codes. It says here, genetic codes have attractions beyond a larger vocabulary. It is this universality of today's genetic code that allows viruses to force cells which they attack to do their bidding, making their viral proteins from their viral genes. A genome that uses different genetic code would be impregnable to such attack. The virus's genes would no longer describe the proteins it needs. Recoding could thus make cells immune to any viral attack. Indeed, there is already work on achieving this in bacteria. And that's one of the programs DARPA's working on, actually.
But wow, TMC, it's like viruses in computer systems, all right? There are people out there that write viruses. And then, certain companies come along with antivirus programs. And then the other guys come along and they build another virus. And sometimes even people that work for the antivirus company in their spare time, just for fun, are writing viruses. So what happens here is if you create a different genetic code, as you just read, that would, I highlighted too much, that would be impregnable to an attack by what was the classical form of life, then all that might happen is, once it now might come along with some other form, and once again we could have an attack anyway. So it's kind of like it's kind of It's a war.
It's kind of like being given a shot, only they already know what the counter to that shot is. So rather than having your natural immune system, for example, they've reprogrammed your immune system so that now it's potentially incredibly vulnerable to some other specific genetic code that then becomes a remote time delay.
Yep.
Kind of thing. I mean, they did say in this article earlier that these people were looking at biotechnology for weapons purposes and then saw great value in making alternative energy fuels, only that fizzled out because it wasn't economically viable.
Right.
So, genetic reprogramming to quote unquote become immunized to something, does that open you up to being incredibly vulnerable?
Not specifically, but if you're going to inject code to immunize someone to something, you can also inject another little bit of code to make a person susceptible to something else that has not yet been released.
That's right. That's exactly right. So you are now the Microsoft operating system that sucks. That's horrible.
You are now at great risk because, a person could become at great risk because they have been modified with an unexploited weakness going forward, and you won't know it. And, we're not fear and doom and gloom and conspiracy theory. And by the way, this show and all of the others that we do here are for entertainment purposes only.
Yeah. I'm just looking at weapons, potential weapons platform. That's really what we're talking about here. I mean, this is the new frontier of technology is
that's right.
Understanding and mastering biology. And when we look at how technology historically has been developed, it's been developed for war.
This new we've looked at two sides of the new frontier. One side of the new frontier is making organic computer circuits. All right. And the other part of it is the idea of being able to program, it kind of goes hand in hand with being able to, make organic computer circuits as well with protein nanowires. But once we have opened this new can of worms of utilizing biomolecules for all this stuff and getting in there and modifying them, it's all about the code. That's where the intelligence is. That's where the structure and organization of any living creature is. Going in there, it's like the final frontier of tinkering. Do you know of a greater final frontier other than tinkering with the very code of life? I ask you. Is there another frontier that's even grander than that one, or is that like it?
I don't know. It depends how mystical or whimsical or out there you want to get. But I mean, we're looking at understanding how to manipulate or manifest in the material space, and they say that that's only 3% of the entire universe. So to say this is the final frontier, I don't think so. So
the thing of it is, the thing that always strikes me so much here was Caroline. It seems like there's a whole lot of a flavor of the scientists defending what they do. They feel a need, or they think that there is, and maybe there is. I'm not saying that there isn't. Once again, it's just my opinion. But Caroline Ajo Franklin is vehement. She's emphatic about, well, we have to fix this. We have to do that. We've got to undo some of these problems. And okay, well, recoding life. All right, I'm going to read the next paragraph. If this sort of recoding could be very helpful to existing biotechnology. Fermentation tanks that never get wiped out by infections, and antibody-producing cell lines that could not harbor viruses would be a great, TMC, this is like, potential poppycock here, because for everything that is being purported to be the potential benefit, there is the opposite side, all right? Fermentation tanks that never get wiped out by an infection. Yes, a fermentation tank where the yeast escapes from the tank and it can't be wiped out by anything. And the yeast takes over the planet, and we're living in yeast world after about 20 years. things can go wrong. Am I crazy? No, I am not. Not at all. All right. Antibody producing cell lines that could not harbor viruses. Well, there is a kind of a balance in nature, though. You know, viruses have been around since, before creation day, and they have existed and coexisted in a balance with bacteria and in a balance with eukaryotic cells, such as human cells. There has been a balance. So to wipe out or to make it antibody-producing cell lines that could not harbor viruses, well, that would be subtracting something out of the natural world that we've been living in for however long your construct of creation or evolution was. All right? There's a problem with that. This is not something we want to do. I know everybody think it sucks when you get a cold, okay? But you know what? Those viruses are there for a reason. They also transfer genetic information from one organism to another. They are almost a kind of like a communication subchannel, if you will. You know what I'm saying?
Yeah, something like this would be applicable if you were going to send people, be it the military or Peace Corps or Red Cross, whatever, right? Send them into an area that's got like dengue fever, yellow fever, malaria.
Sure. Make them immune. Okay.
Yeah.
Cool.
And you could do something like this with the time to live, or reverse it when you come back, kind of a thing. I mean, I can see where there's some marketability here with some of this kind of technology. I can see that. I do. But I'm going to sit back on my haunches there, and I'm going to say viruses are a natural part of the world. There's something that they do, that they've been doing, along with bacteria and along with yeast and along with E. coli and along with humans and monkeys and insects and every other creature. They've been doing it for a long time, and it's been working. So what happens in nature is if you take a balanced system like that and you take something out, now you've upset the balance. This is just common sense, right? So you take out, I don't know, whatever it is, some kind of insect. And all of a sudden, if you take out that insect and that insect has been eating something, now you get too much of whatever it was eating because it's not eating it anymore. And now another creature will start taking advantage of the imbalance in the system. All right. So I think that this statement right here, cell lines that could not harbor viruses would be a great boon. I think that is a completely fallacious and misguided statement. And I will stand on my position on that. All right. Let's expand it to bacteria. Let's say human cells were redesigned in such a way, or were reprogrammed by some sort of a delivery of messenger RNA, hint, hint, hint. I don't know how that might be, but let's say that they were reprogrammed so that they would not harbor or have the potential for bacterial infection. Well, that's upsetting the balance. And guess what? There are more bacteria in our bodies coexisting with us than there are actual human cells. So, upsetting the balance once again. Is it me, or is it science gone too far? I think there's definitely a balance. There's definitely a balance.
So it goes on. It is possible to imagine, I think I forgot the time. We're out of time.
Yep.
It is possible to imagine changes in the way codons code for amino acids, so radical that parts of synthetic biology become a separate creation. I don't know. where are we going to put that separate creation, TMC? Is that going to be like here? Is it going to be one that outcompetes us? Is it going to displace us? Am I just being afraid? Should it be something that should be tried on another planet? I don't know, man. What do you think?
Yeah, I think it's
a brave new world.
It's exactly what I was going to say.
Well, it's an amazing world. I love the comments that come in, and one comment, it's called Let's Play God. Yeah, it's just, I don't know. I don't think that that's something that we should be doing, but I see no divergence from the path of doing it. All right. We're out of time. Thanks, everybody, for joining us this week. This is a beginning foundation of what we're going to show you, actual stuff coming up. We will show you, we will feature more video from more people who speak about what they're developing, as well as more documents and diagrams of what this all means going forward. We are going to change we're going to start, not we personally, but we're going to start building new life forms. And I don't know where that's going to go. TMC, thanks for being here with me. Do you want to take us out?
Sure. Good night, and come back. We're gonna circle back around to how this interplays with humanity. We'll get into the different genetic manipulation technologies that they're looking at to interact with the human organism. So that's where this is really going, is to look at some of those technologies. So more as time permits. Good night.